So the purpose of this video presentation is to look at the journal article summaries that were provided by some students. So I'm going a little bit back here. I'm going to go over a journal article summary that was provided by Maria before we took our break from actual lectures. This article looked at the effects of cold water immersion after training sessions, and this was during 6 weeks of sprint interval training, and they looked at the effects of the cold water immersion after each training session on changes in the muscle sodium potassium transport proteins in the muscle fibers. So if you recall from earlier in the course, you have sodium potassium pumps in your sarcolemma or your muscle fiber membrane, and they're important for restoring the muscle fiber electrical charge after exercise. So during exercise, you have sodium that rushes into the muscle and potassium that leaves. And the sodium potassium pump acts to get those ions back to their normal places. So after exercise, the sodium potassium pump pumps potassium back into the muscle fiber and pumps sodium out, and that will restore the resting membrane potential of the muscle. So while those ions are in imbalance, especially with the potassium, so recall that you get hyperpolarization during muscle contraction when potassium leaves the muscle. You need activation of that sodium potassium pump to get that potassium back in to restore your muscle membrane potential back to normal. If you can't do that quickly, then you'll have trouble generating another action potential, and you'll have trouble essentially you'll have trouble depolarizing the muscle membrane if you can't get that recovery of the potassium back into the muscle. So anyways, this study looked at the effects of 6 weeks of training on this sodium potassium pump and looked at whether cooling after exercise actually affected the expression of that sodium potassium pump in the muscle fiber membrane. So a couple of points that were made in the introduction of the article, and I'm going to go to the whiteboard for this, is recall that when Horea presented her article, it showed that protein synthesis was impaired during a resistance training program if the leg was placed in cold water after each training session. So it looked like post-exercise cooling of the muscle with resistance training actually impaired protein synthesis. And in Maria's paper, in the introduction, they mentioned that satellite cell activation was impaired in previous research where there was post-exercise muscle cooling. So I'm just going to go over the importance of satellite cells. So if you have your regular muscle fiber here, it is surrounded by satellite cells that are outside the muscle fiber membrane. And what normally happens with resistance training, so this is with weeks and weeks of resistance training, you get proliferation of those satellite cells, so they actually increase in number. And then those satellite cells, they actually get incorporated into the muscle fiber as nuclei. So this would be one nucleus. Within the muscle fiber, a muscle fiber is a unique cell in that it has multiple number of nuclei. And those nuclei are important for protein synthesis. So that's where your protein synthesis essentially starts. So with regular resistance training, you'll get an increase in satellite cell number, but the satellite cells will also get incorporated into the muscle fiber and turn into new myonuclei. and that enhances your capacity for protein synthesis. So that's the normal response to resistance training. And according to Maria's paper in the introduction, they mentioned that this whole response is impaired if you cool the muscle after your resistance training sessions. Now, essentially what Maria's paper found was that the sodium potassium pump it's actually increased with training, and it was increased more so in the fast twitch fibers, probably because of the type of training they used. So they used a sprint interval type of training, so it's high intensity. You're most likely recruiting those fast twitch fibers, and you're seeing greater adaptation in those fast twitch fibers. So that's the normal response you get to training, is you get an increase in the sodium potassium pumps in the membrane of the muscle fiber. And that's going to help your muscle to recover after exercise. It's going to help to pump potassium back into the muscle and restore your resting membrane potential. So essentially with Maria's paper, they found that post-exercise cooling had no effect on the changes in expression of the sodium potassium pumps. So one thing I want to point out that Maria's paper did measure is they measured mRNA levels and protein levels of the sodium potassium pumps. So this is kind of a good review of how proteins are expressed in a muscle fiber. So I'm drawing the nucleus a little bit bigger here. And this whole thing is a muscle fiber. And with Maria's paper, they assessed mRNA levels, so mRNA expression that corresponds to the sodium potassium pumps. So in your nucleus, you have DNA, and that encodes for all the proteins for your cells. When you have something like an exercise intervention, some of those proteins will be expressed or increased in expression. So in this case, it's the proteins for the sodium potassium pump. First of all, you get a portion of that DNA that's copied. So some of the bases get copied onto what's called mRNA. So that's mRNA. And that process is called transcription. So this is transcription. So you're just copying a portion of the DNA onto mRNA. And then the mRNA, it takes that code for a specific protein outside the nucleus, and it goes to your ribosomes. This is ribosome. And that's where you get the actual protein synthesis. So you get amino acids that come into the ribosome that correspond to a message on the mRNA. And it arranges those amino acids in a specific sequence to make a specific protein. So in this case, it was making the protein that corresponds to the sodium potassium pump. So this process on the ribosome, where you get the message from mRNA translated to make a new protein, that process is called translation. So this is a little bit of a review. I think I went over this a little bit earlier. So with Maria's paper, they measured the mRNA levels for the expression of the sodium potassium pumps, and they did that after the first exercise session. So that's to kind of get a handle on what's happening early in the adaptation to exercise. And then after the 6 weeks, they assessed the protein levels of the sodium potassium pumps. So mRNA is expressed earlier, and then the whole protein would be expressed a little bit later. So that kind of summarizes the important points that I wanted to make from Maria's paper. So the other journal article summaries that I posted were, I'll just have to get my computer going here again. So another one that was posted was JC's article, and that one was on the effects of menstrual cycle phase on peak fat oxidation during a graded exercise test. So essentially what they're doing here is they're estimating fat oxidation during a graded exercise test, and they're looking at the peak fat oxidation during that graded exercise test, and they've assessed it at different phases of the menstrual cycle. So this is a bit of a review of what we covered in class. It's how estrogen changes during different phases of the menstrual cycle. So here you have estrogen, and during the menstrual cycle it would go something like this. So what you have here is you have your, this would be around day 14. This is day 0, this is say day 28. This is your follicular phase. And this is your luteal phase. And recall that some previous research showed that with higher estrogen levels, you get a greater fat oxidation during exercise. And so in this paper, they've hypothesized that they've assessed fat oxidation during this graded exercise test. Essentially, they've assessed maximal fat oxidation. They've measured it at 3 different periods, 3 different time points in the menstrual cycle. So they've measured it during the late follicular phase, when you'd have your highest estrogen levels. And then they've assessed it in the mid follicular phase, when your estrogen levels would be relatively low. And they've also assessed it during the luteal phase, when your estrogen levels would be relatively high. And essentially they found that there was no difference in the maximal fat oxidation levels, despite these changes in estrogen. So, from this study, you can say that the changes you get in estrogen during the menstrual cycle, they don't really affect your maximal fat oxidation during exercise. Another point I wanted to make from Jace's paper is they've assessed free fatty acids and glycerol from the blood. And these come from fatty acid breakdown. They come from triglyceride breakdown. So again, this is a bit of a review. So if you had your adipose tissue here, your adipose tissue stores your triglycerides. So during exercise, your triglyceride gets broken down to glycerol and 3 free fatty acids. So often fatty acids and glycerol are measured in the blood during exercise to get an idea of your rate of lipolysis, or how much your triglycerides are breaking down during exercise. Now, scientists generally like the glycerol measurement because this sticks around in the blood longer, whereas the fatty acids, they can either go to muscle to be taken up and oxidized, but sometimes they can be taken up by the adipose tissue again and then re-esterified. So the free fatty acid levels are kind of less reliable as a marker of triglyceride breakdown during exercise. And glycerol, I would say, is probably a more reliable marker, a little bit more difficult to assess, but that's considered a more reliable marker than the change in free fatty acids. And at this point, I'm going to take a bit of a break, and I'm going to post the journal article summaries for 2 more journal articles that were sent to me on the response of exercise to cold again.